Good morning, grade 12s. My name is Someleze. My surname is Makubalo. I'm here to talk to you guys about meiosis. So this is a new topic that I'm introducing to you guys. Meiosis is all about cell division. So if you talk about meiosis, all that you need to know is that the cell divides. That's all. So now I'll start by talking about interphase. Interphase, as you can see here, I've written that chromatin network. So these are chromatin network. These are chromatin network. All this that you see here is chromatin network. What happens to interphase? One thing that you need to know is that in interphase, just basic information, there's nothing much that you need to know. But you need to know that in interphase, DNA replicates and double itself. So basically, N chromosomes start to what? To form. So in interphase, it's whereby the DNA replicates itself, and then you start to see DNA strands. Basically, as you can see, this looks like a wire or a rope. So DNA will start to will double itself, and then you will have something like a rope, which are strands of Strands of DNA, but we call them chromosomes. So chromosomes start to form in interphase, but they are not really visible when it begins. It starts by being chromatin network, and then it replicates itself. So now, what I want us to focus on today is that we are going to deal with chromosomes and chromatids in this phase. Before I even get to prophase 1, I want to first explain to you guys this. This, we call it a chromatid. This is a chromatid. A chromatid. Chromatid. When it's just one strand, it's called a chromatid. But once you start to see, this is then called a chromosome. A chromosome. This is a chromosome. A chromosome is made up of two chromatids that are joined together by By a centromere. So a centromere joins chromatids together and forms a chromosome. So if we are saying in interphase, it's a DNA replication that end up forming chromosomes. That means at the end we'll have strands that are joined together by a centromere that will form a chromosome. So that is a very important information that you need to know, because sometimes textbooks, they can confuse you. Sometimes you'll hear that they will call a chromosome a chromatid. So, but I want you to understand this, that a chromatid is a half of a chromosome. Then once you talk about a chromosome, you're talking about two joined chromatids that are joined by By a centromere. They form a chromosome. So if you talk about a chromosome, you are talking about two joined chromatids. So all that you need to know in interphase is that you have chromatin network. These are chromatin networks, and then the DNA replication, and they double itself. So DNA replicates and double itself. That's all that you need to know about prophase 1. Then now let's move on to, interphase. Let's move on to prophase 1. What happens to prophase 1? I'll try to explain all these diagrams. They represent prophase 1, but I wanted to show you guys from what stage to what stage, and what's the final stage of prophase 1. So this is what happens. The chromosomes, we call them homologous chromosomes. Why are they homologous? Because they are the same. You know the word heterogeneous and homozygous and all those things. So when something is homo means the same. So if, as you can see here, we have two chromosomes that are drawn with a blue color. So those two chromosomes, they are homologous. Let's assume that these two chromosomes, they're coming from paternal. What is paternal? Paternal means father. So we have two chromosomes that are coming from the paternal that are homologous. And then we have also these two chromosomes. I've also written them as homologous chromosomes. They say they are coming from maternal. Maternal means mother. So these two chromosomes, I want you to understand this. Normally, in a normal setup, it's 23 from the father, it's 23 from the mother. But in meiosis, when we use in grade 12, when we talk about meiosis, we don't take all the chromosomes. We just take 2 from the father and 2 from the mother to show an overall illustration of what really happens in real life. So we have 2 from the maternal and paternal, we have 2 from the maternal. Now, I want you to see what happens. Remember, I'll make this example. You have seen a child that looks like the father, and sometimes you'll see a child that looks like the mother. Sometimes you'll see a child that has features of the father and features of the mother. For instance, maybe the face looks like the mother and the child is a boy. And maybe the lower body, it looks like the body of the mother whatsoever. But all that I want to explain to you is that the genes that come from the father and the genes that come from the mother, they mix together. And after mixing together, they divide. After that division, then they form, or rather they have all the characteristics that will be passed on to a child. So before a child is formed, there's a lot of mixing that takes place. So what I want to explain to you is that, as you can see that this is from the mother and then this is from the father, and then you have a nuclear membrane. That's the nuclear membrane. Now let's move on to the second diagram. Remember, a chromosome with 2 chromatids. So a chromosome is actually formed together by what? 2 chromatids. Remember what I was explaining and what I drew also on this side of the board. So here we have the maternal and the paternal. Now let's move down. This is what happens. The chromosomes from the father and the chromosomes from the mother, they mix together. They, as you can see here, the chromosome which is red from the mother is crossing with what? With the chromosome from the father. And that point of crossing over, that point whereby they join, we call them what? Chiasma. That is called what? Crossing over. The point whereby the chromosomes are crossing each other is called crossing over. But now that point of intersection is called chiasma. So now what happens? Why do we have crossing over? It's whereby the genes, the genetic material of the mother is mixing together with genetic material of what? Of the father. Then I would love to use a lot of examples on this. Let's assume that the father is sharing a gene to the mother and the mother is sharing the gene to the father. That combination gives birth to a child that has certain features that are coming from the father and certain features that are coming from the mother. The other word that I would love to introduce to you guys here is variation. This whole process is all about variation. It is to give to a new different, sometimes give to a new different child, or rather, I have to start afresh.